So far, I've described one situation that can give rise to non-Mendelian inheritance patterns. More specifically, I talked about allelic relationships that are non-Mendelian. So things like codominance, incomplete or partial dominance, or lethal alleles were the three main examples that I've described to this point. So what I want to do now is describe a second mechanism or a second situation that can give rise to non-Mendelian inheritance patterns. And this is a phenomenon that we call epistasis. So first, let me just define epistasis. It's where one gene will act to mask or modify the expression of a second gene contributing to the same trait. And when you have epistasis and you're performing genetic crosses, there is the potential, at least, to observe non-Mendelian inheritance patterns or phenotypic segregation patterns in segregating populations. So we're going to go through several examples of this, but I want to again indicate how it deviates from the Mendelian conditions. So let me describe the Mendelian conditions again. So for epistasis to occur, first of all, you have to have a dihybrid situation. Okay, so again, the Mendelian dihybrid was such that the two genes that were being analyzed in a given set of crosses were each determining the different phenotypes, determining the expression of two different traits. So again, by hybridizing two pure breeding parental types that contrast in their form of expression for a given trait, generating an F1 dihybrid, either selfing or intercrossing that F1 dihybrid to produce an F2 segregating population, as shown here again, using Punnett square analysis. What you can derive are the Mendelian observations. So phenotypically, Mendel saw in his F2 populations of dihybrid cross a 9 to 3 to 3 to 1, 4 different phenotypic types occurring in a ratio of 9 to 3 to 3 to 1. 16ths were dominant for the two traits. 3/16ths were dominant for the first trait and recessive for the second. 3/16ths were recessive for the first trait and dominant for the second. And then 1/16th of the F2 population were recessive in their expression, their form of expression for the two traits. Okay, so again, what are the Mendelian conditions that give rise to that specific Mendelian segregation pattern? So you have each of the two genes controlling different traits. You have dominant recessive alleles in each of those two genes. And then lastly, you have independent assortment. So again, that's what allows you to predict, when you have those conditions, those Mendelian conditions, you always expect to see an F2 population that will segregate 9 to 3 to 3 to 1. So again, the information derived from this Punnett square is both information about a predicted phenotypic segregation pattern as well as a genotypic segregation pattern. So again, from this Punnett square, what you can determine is that there's a total of 9 different genotypes that are represented in that F2 population, and they occur in a ratio of 1, 2, 2, 4, 1, 2, 1, 2, 1. Okay, so actually all of this information, both the genotypic ratios and all of the different genotypes that occur in that F2 population and the phenotypes and their relative proportions can be summarized over here in this little table or chart to the far left. So what do I mean by that? Why does that summarizes all of the information describing that F2 population, both genotypically and phenotypically. So let me just show you on this slide here. So again, here's all of the same information, phenotypes in the F2 population of a Mendelian dihybrid cross, and all of the predicted genotypes and their relative proportions as well. So this group here, 9, where I have G, big G, empty slot, big W, empty slot, and so on, what you can see is, that represents a phenotype that summarizes this phenotypic information. You're dominant for both the G and the W trait, right? The capital G and capital W. But in addition to that, it's also representing all of these potential genotypes. So, all of these genotypes over here within that phenotypic class, big G, and then for the second allele at the G gene, it can be either big G or little g, right? So that's why we leave it empty. It can be either one of the alleles, big G or little g, in that second slot. Same thing with W. You can see, all of these 4 genotypes have a big W or capital W allele, but for the second allele, it's either big W or little w. So it could be either one. So we just leave it empty. So again, this designation here, 9, G dash W dash, summarizes all of this information for that phenotypic class. Summarizes the phenotypes, dominant, and summarizes all 4 of the possible genotypes within that phenotypic class. So that's true here, for the next phenotypic class, 3/16 that are dominant for G and recessive for W, right? Homozygous recessive here. You can see these 2 genotypes, your homozygous recessive for W, you have one big G allele, and then the second G allele can be either big G or little g. So again, that summarizes all of that information. Same thing down here for this phenotypic class. 3/16 are going to be recessive for G. You see homozygous recessive for G for these 2 genotypes. And then for the W gene, all these 2 genotypes all have big W. And then for the second allele, it can be either one, either big W or little w. And then lastly, the last phenotypic class, again, is going to be homozygous recessive for both G and W. And so therefore, phenotypically, again, it's going to be recessive. So this table or chart or whatever you want to call it summarizes all of this information in this Mendelian F2 population derived from a dihybrid cross. So it's this information that's going to be varied when you have epistasis. So epistasis, again, is when you have 2 or more genes that are responsible for determining a single trait. So that's non-Mendelian. So if you're doing a dihybrid cross and the 2 genes determine the final expression, phenotypic expression for a single trait, you have the potential for epistasis. And there's many different ways that those genes can interact. So again, one gene, when you have 2 genes that are contributing to the ultimate final phenotypic expression for a single trait, one gene can act to mask or modify expression of the second gene. Okay? So I'm going to go through several examples of that. There's many different forms of epistasis, many different ways that these genes can interact with each other when you have 2 or more genes determining a single trait, the expression of a single trait. So again, in the early 1900s, as I said, researchers and breeders and so on were doing genetic crosses with all different types of organisms. So here's one example. So in the mouse, so they were trying to determine the genetics, the underlying genetic basis for coat color in the mouse. So what they would do is they would take 2 pure breeding parental types that contrasted in their coat color. They would generate an F1 hybrid, and then they would intercross the F1s to produce a segregating F2 population. And then based on the segregation pattern, they would build a genetic model. That's how you do this. This is how you do, you build genetic models. You look for recognizable segregation ratios. So in this case, when they did this experiment, they saw something that was unexpected, something that was actually non-Mendelian. So in that F2 population, they saw that 9/16 of the population had an agouti coat color, 3/16 had a black coat, and 4/16 had no color, white or albino fur color. So the phenotypic segregation ratio in that F2 population was 9:3:4. So Mendel never saw that phenotypic segregation ratio in any of his crosses. Okay, so they could repeat this, the cross over and over again. They always got the same outcome. So it was, the outcome was predictable. I mean, in terms of repeatability, but they couldn't explain it based on an understanding of Mendelian genetics. So there must be something non-Mendelian going on. There's clearly a genetic basis, a biological basis, because of the repeatability, but there had to be something, going on, a mechanism acting that was acting in a non-Mendelian fashion. So they looked at that ratio and they said, well, actually, that looks like a variation of a 9 to 3 to 3 to 1, right? If you take those four phenotypic classes and you group two of them together, a 3 and a 1, you can come up with a phenotypic segregation ratio of 9 to 3 to 4. So, the light bulb went off and they said, okay, well, maybe there's, something unusual going on here in terms of genic interaction. In other words, it was the phenotypic ratio of 9 to 3 to 4 was a deviation from 9 to 3 to 3 to 1. So that suggested that there's two genes involved in determining the one trait of coat color. So with that recognition, they built their model. This is what they proposed. They said that there's two genes that are responsible for determining coat color in the mouse. So I've defined those genes as the A gene and the B gene. I've assigned dominant recessive alleles at both A and B, and then I've assigned phenotypes that are conditioned by those two allelic alternatives at each of those two genes. Okay, those alleles, the big A is dominant to the little a, the big B is dominant to the little b. And then the last component of this model, or another component of the model, is the idea of independent assortment. Okay, then the last component of the model is where they proposed a specific form of epistasis or genic interaction. Okay, so what did they determine in terms of a genic interaction? So what they did is they looked at, they used this chart, okay, that I showed you earlier. Okay, so what they said is that here's the 9/16 of that F2 population that are agouti. Here's 3/16 that are black. And then if you group these two phenotypic classes together and all three of those genotypes together into a single group, you would then have the 4/16 of the population that were albino. So what they were saying is that these four genotypes that fall within this phenotypic class of dominant were agouti. These two genotypes that fall within this phenotypic class of dominant recessive were black. And then these three genotypes, homozygous recessive A, homozygous B, big B, or heterozygous big B, little b, and then homozygous recessive A and B, they said that all three of those genotypes produce the same phenotype of albino. That would then give you a 9 to 3 to 4 phenotypic segregation ratio. So what's the nature of the epistasis? What's happening is that if you have a dominant A allele, that allows for color, pigment to accumulate in the hairs of the fur. And then the color is determined by the second gene, the B gene. So if you're big B, based on this model here, so A allows for color. If you're big B, you're going to have an agouti coat color. And if you're homozygous recessive B, you're going to be black. So this would be color, agouti, color, black. So 9 to 3. What about this last phenotypic class of albino? So if you're homozygous recessive for A, based on the model, you can't get any color. So it doesn't matter what alleles you carry at the B gene, you're still going to be colorless, white or albino. So homozygous recessiveness at A is masking or modifying expression of the trait determined by the B gene, which is color. So it doesn't matter what allelic combinations you have at the B gene. If you're homozygous recessive A, you're going to be colorless regardless. So this is a form of epistasis that we call recessive epistasis. Homozygous recessiveness at the A gene is masking or modifying expression of the B gene. And that type of epistasis will give rise to a 9:3:4 phenotypic segregation ratio. Okay, so that's one type of epistasis, one type of genic interaction. There are many others. So here's another example. So here's a fruit color in squash. Okay, so plant breeders were trying to work out the genetics of color, fruit color. So you take two pure breeding parental types, contrasting their form of expression of the squash, generate an F1 hybrid, intercross yourself to produce a segregating F2 population. And then based on that segregation pattern, you can, build a model as long as it's a repeatable pattern, segregation pattern. So what they saw, again, was something that was non-Mendelian. What they saw in this particular set of crosses in that F2 population is that 12/16 of the plants produced white fruit, 3/16 produced yellow, and 1/16 produced green. So a 12:3:1 phenotypic segregation ratio. Okay, that's non-Mendelian. But they recognize that, again, as a variation of 9:3:3:1. So if you combine the 9 and the 3, you get 12, and then 12:3:1. So again, what they proposed here is that the trait is determined by two genes, and they're interacting in some way, and an epistatic interaction to cause this non-Mendelian pattern, segregation pattern. So they built a model. So here's the model. Again, two genes, the A gene and the B gene, they both contribute to expression of the same trait, fruit color. There's dominant recessive alleles at A, big A and little a, big B and little b. They assigned phenotypes conditioned by each of those four alleles. They included independent assortment in their model. And then lastly, they had to determine the type of epistatic interaction that's giving rise to this non-Mendelian phenotypic segregation ratio. So again, here's your little chart. So how do you get a 12:3:1? You group these two Mendelian phenotypic classes together, and that's 6 different genotypes. And you say that because of the epistatic interaction, all 6 of those genotypes produce the same phenotype of colorless or white, a white fruit. These two genotypes that fall within this phenotypic class, the 3/16, would be the yellow, would be the genotypes that determine the yellow phenotype, and then homozygous recessiveness at A and B, 1/16 of the population would produce the green phenotype. So what would be the nature of the epistatic interaction here? It looks to me like it must be homozygous, or I'm sorry, a dominant allele at the A gene is acting epistatically on the B gene. So it's only when you're homozygous recessive at A can you actually get a colored fruit, and then the color is determined by the B gene. So this is actually how they built their model. They said dominant A produces no color, colorless fruit, or a white fruit, and that's the allele that acts epistatically. When you're homozygous for the recessive allele, you can get color, and then color is actually determined by whatever allelic combination you carry at the B gene. So again, dominant A acts epistatically on B. It doesn't matter what alleles you carry at the B gene, you're going to be colorless or white. When you're homozygous recessive for A, then you can get color. And again, the color depends on whatever alleles you're carrying at the B gene. So this model and this particular form of epistasis would explain a 12:3:1 phenotypic ratio. So this is what we call dominant epistasis because it's a dominant allele that's acting epistatically on the second gene. The previous example, it was a homozygous recessiveness. It was acting epistatically on the second gene. Okay, so we can go on. Here's another example. So remember when Mendel did his work, he one of the traits in P that he was interested in was the genetic basis for flower color. So that's analyzed in his monohybrids, produced an F2 population that segregated 3 to 1. Okay, so a single gene trait, dominant recessive alleles, very predictable. So subsequently, other plant breeders were doing other experiments with different strains of the P, different genetic backgrounds, and analyzing inheritance of flower color again. And they were able to generate an F2 population that segregated not 3 to 1 like Mendel, but 9 to 7. So 9/16 of the F2 population had purple flowers, and 7/16 had white or colorless flowers. So it's the same trait that Mendel analyzed, but now you're getting a completely different segregation ratio in that F2 population. So how do you explain that? What's going on? Okay, so again, they looked at the 9 to 7. They said, well, that looks to be a variation of 9 to 3 to 3 to 1. If you combine the 3 to 1 together, you get 7. So again, they proposed a model that incorporated and described a third type of epistasis, or a third type of epistatic interaction. So again, they define 2 genes, the A gene and the B gene. They both contribute to this determining the expression of a single phenotype flower color. They said there's 2 alleles, dominant and recessive at both A and B. The dominant allele produces purple for both genes, and the recessive allele produces white or colorless flower for both genes. And then they, again, their model included the idea of independent assortment. And then they had to, top it all off by describing the particular form of epistasis to complete their model. So here's the chart again. This is the Mendelian situation. So again, as I said, if you group all 3 of these phenotypic classes and all 5 of those genotypes together, and then build a model that proposes they all are capable of producing the same phenotype of white, then you would end up with a 9 to 7 overall phenotypic segregation ratio in that F2 population. So it's only these 4 genotypes up here that would produce the phenotype of purple flower. Okay, so what's the nature of the epistatic interaction here? Well, it seems to be that you need a dominant allele at both A and B in order to produce a colored flower, a purple flower. If you're homozygous recessive at either B or A or both, you're going to produce a white flower. So again, based on that, it appears that homozygous recessiveness at either A or B acts epistatically on the second gene. So homozygous recessive B will act epistatically on A. So homozygous recessiveness at B masks or modifies expression of the A gene. It doesn't matter what alleles you carry here. You're still going to get a white flower. So same thing here, homozygous recessiveness at A masks or modifies expression of the B gene. It doesn't matter what alleles you carry at the B gene, you're still going to produce a white flower. And then, of course, if you're homozygous recessive for A and B, you're going to produce white as well. So this particular form of epistasis is what we call double recessive. So homozygous recessiveness, either A or B, acts epistatically on the second gene to then give rise to an overall phenotypic segregation ratio of 9 to 7. So the point here as well is this idea that Mendelian crosses and Mendelian analysis was very simplistic. So the idea that a single gene is responsible for determining a single trait. But in reality, you look at any trait in any organism, And almost always, I mean, if you do enough work, you're going to be able to identify more than one gene that ultimately contributes to the expression of that particular, of a single trait. So it's not uncommon to observe epistatic interactions because almost always for any given phenotype, any given trait, you have two or more genes that contribute to the expression of that trait. And it's very rare that you would have a single gene responsible for a single trait. It's not to say you can't have genes that are major contributors to the expression of a single trait, but it's rare that there's a single gene that determines the ultimate expression of a single trait. So again, the point is that epistasis is a fairly commonly observed in genetic process. So there's one other, in this table here, we've done the 9 to 3 to 4, we've done the 12 to 3, there's the 9 to 7. You can do other crosses where you have another form of epistasis that gives rise to a 9 to 6 to 1 phenotypic segregation ratio in an F2 population. That again is a variation of a 9 to 3 to 3 to 1. 13 to 3 is another type of epistatic interaction. Again, the 13 to 3 is a variation of a 9 to 3 to 3 to 1, and so on, 10 to 3 to 3. Here's the last one I want to describe, the 15 to 1. So this is shepherd's purse. So it's, weed or an ornamental, I guess, for some. And they were working out the genetics of the shape of the seed pod or the seed capsule. They took two parental types that contrasted in their form of expression for the trait, generated an F1 hybrid, generated an F2 population, selfing or intercrossing, and they saw 15 to 1. So again, that's clearly non-Mendelian. Mendel never saw that type of ratio in an F2 population. So they had to explain it. So again, they recognized the fact that the 15 to 1 seems to be a variation of a 9 to 3 to 3 to 1. So they built their model, again, a 2-gene model, dominant recessive alleles, independent assortment for the alleles of the 2 genes. And then lastly, they invoked a new type of epistatic interaction. So here's the model. The A gene and the B gene, determine pod shape, the same trait, dominant recessive alleles at A and B. They said A produces a triangular shaped seed pod. Same thing for B. And then the recessive alleles at A and B produced an ovoid shaped seed pod. Okay, so then the last thing they had to determine was the nature of the epistatic interaction. So you go to this chart again. So to produce a 15 to 1, you have to group all 3 of these phenotypic classes together. And all 8 of those genotypes within those 3 phenotypic classes. And you propose a model in which all 8 of those genotypes produce the same phenotype. It's only when you're homozygous recessive for both A and B do you get the alternative phenotype, ovoid. So what type of epistatic interaction gives rise to this 15 to 1 ratio? It's what we call double dominant. So all that you need is a dominant allele at A or B to produce the phenotype of triangular shaped seed pod. So again, a dominant, so it's dominant epistasis, but both the A and the B gene are acting epistatically on each other. So in other words, if you have a dominant A and a dominant B, you're going to produce triangular. If you have a dominant A, that's going to act epistatically on the B gene. It doesn't matter what alleles you carry at the B gene. If you carry just a dominant B allele, again, that will act epistatically on the A gene. It doesn't matter what alleles you carry here. And it's only when you're homozygous recessive for both A and B that you produce the ovoid shaped seed pod. So this is a form of epistasis that we call double dominant. epistasis. Okay. So there is one other example in this table here, and I just want to mention it briefly. So here they were working with the flower beetle. So believe it or not, they've done genetic analysis of the flower beetle. Actually, it's a model organism that's been used in population genetic studies. So they were just trying to work out, the color of the bug. So you take two pure breeding parental types, have different forms of, pure breeding for different forms of expression of the trait, generate an F1 hybrid, intercross the F1s to produce a segregating F2 population. So that's what they did, and this is what they saw. They saw a phenotypic segregation ratio of 6 to 3 to 3 to 4. Okay, well, that's clearly non-Mendelian, but it doesn't look like it's an obvious variation of a 9 to 3 to 3 to 1. So the suggestion is that it's, it's probably a 2-gene trait. There's some form of epistasis going on, but there must be something else going on as well to give rise to this really strange segregation ratio in that F2 population. And actually what it is that, again, I'm not going to go into the specifics of this. You can kind of play around with it yourself, or we can discuss it at another time. But basically what, you have two genes that are contributing to color, and the alleles at one of those two genes act dominant recessive relative to each other. And then at the second gene, they act in a co-dominant manner. Okay, so there's actually two non-Mendelian conditions in this particular example to give rise to that really unusual phenotypic segregation ratio. So there's also epistasis going on, there's also independent assortment and so on, but one of the genes carries dominant recessive alleles that contribute to color, and the other gene carries codominant alleles. Okay. So there's one other example I want to give to demonstrate this idea of epistasis, and it goes back to something we've discussed previously when we talked about non-Mendelian allelic relationships. So we described the ABO blood typing system in humans, and how in that 3 allele series, A and B are dominant to O, but they're co-dominant to each other. So here was an unusual observation in a family from India. So here they had one of the parent. This is a pedigree. We'll talk about pedigree analysis in a future lecture, but I'm sure you have maybe general familiarity here. But at the top here we have the two parents. The circle is the female, the square is the male. So the mother had an AB blood type and the father had an A blood type. So what do we know? Genotypically, the father can be either one of two types, either homozygous A or heterozygous, and the mother must be heterozygous for A and B. So those two individuals produced a child that had the O blood type. So based on our understanding of the I gene and the 3 alleles of the I gene, that's impossible. There's no way that those two parents with their particular genotypes could produce a child with an O blood type. Okay, then in addition, that child grew up, uh, uh, married, I assume, intermated, had children, 3 children, and again, this individual has the O blood type, and the father, the husband, had the A blood type, and yet they were able to produce children that expressed either the B type blood or the AB type. So where's that B coming from? It couldn't be present in an O genotype, or it couldn't be present in an A genotype as well. So clearly something unusual is going on here. And what they were eventually able to determine is that there was epistasis occurring. So actually, as I said, it's not uncommon to, the ultimate determination of the blood type within the ABO blood typing system actually relies on, more than one gene. There's actually a second gene that plays a role in determining your particular phenotype for this blood type system. So this individual, this is what's called, if you have, an A individual crossed with an AB and you produce an O, an unusual outcome like that, this is what we call a Bombay phenotype, and it's explained by epistasis. So how does it work? So there's actually a second gene that determines this blood type, and it's called the H gene, and it's over down here in the lower left. So the H gene, there's two allelic alternatives, and they act in a dominant recessive manner. Okay, so there can be three genotypes at the H gene: homozygous big H, heterozygous homozygous recessive. The dominant H allows for the production of something that we call substance H. And then the recessive allele, you don't produce substance H. Okay? So again, if you're homozygous, big H, or heterozygous, you're going to produce that substance H, and it's going to be present on the surface of your red blood cells. So it's that substance H that is modified by the gene products of the I gene to then determine a particular phenotype of A, B, O, or AB. So in other words, the I superscript A allele adds a sugar, the A sugar, onto that substance H to produce the A phenotype. The I superscript B allele adds a B sugar to that substance H to produce a B phenotype. And if you carry both alleles, you'll be AB. Both sugar types will be added to substance H in those individuals. And if you're homozygous for the O allele, the O allele doesn't add any sugar to substance H. So you'll be of the O blood type. Okay, so for instance, here's an example here. You're heterozygous at the I gene for A and B, and you're carrying a dominant H allele. So this will produce substance H, and then it'll be modified by these two different sugar types to produce the AB phenotype. Okay, same thing here. If you're heterozygous at H, and you're homozygous recessive for the, this is the allelic designation sometimes used for the O allele, little I. So this would be the O phenotype, O blood type. However, what if you're homozygous recessive for H? If you're homozygous recessive for H, then you never produce substance H, and then it doesn't matter what allelic combinations you carry at the I gene. There's nothing there to add a sugar to. And therefore, if you're homozygous recessive at H, that will act epistatically on the I gene such that you will always produce, the Bombay phenotype or the O blood type. Okay, so the H gene, homozygous recessiveness at the H gene, acts epistatically on the I gene. And that's what's going on here. So what that means is that in order for this individual to be homozygous recessive for H, each of these two parents up here had to be heterozygous for the H gene. So they carried a dominant allele, so they still had a blood type as determined by the I gene. But down here, this individual is homozygous recessive for H, so it doesn't matter what allelic combinations you carry at the I gene, you're still going to be O blood type. And then when this individual mated to produce offspring, again, remember this individual still carries the B allele from this parent here, from the maternal parent, so that individual can transmit that to offspring. Okay, so that's the explanation, epistasis occurring within the ABO blood typing system that explains this unusual Bombay phenotype.